Adaptationism. The first speaker will be Alessandro Minelli. His title is Individuals, Hierarchies and the Level of Selection, a chapter in Gould's evolutionary theory. Please.
Thank you, Marco. Morning to everybody. Despite this 8 minutes or so of delay, let me spend a couple of words to give context before introducing you into some technicalities of biology. It's amazing to see how many people are gathering in this room and in the other room to think about evolution. 3 years ago, 2009, the year of the celebration of Darwin's anniversary, there was a lot of initiatives in Italy, and in particularly Telmo Piovani was extremely active. He told me that he was invited to give some 50 lectures along the year. And other people in this room, Marco Ferragutti, myself, and others, were involved in a similar number of events. This demonstrates that in this country there is a lot of interest in evolution. On the other hand, in the same year, 2009, the University of Torino hosted one of the editions of the Congress of the European Society for Evolutionary Biology, a big event attended by some 800 people. But among these large number of young and not so young scientists, and excluding the local organizers, the Italian people attending this meeting were 15 or so. And the majority of these 15 are also here in Venice for this event. Why this difference? I leave this question floating in the air. They will perhaps give us a way to ask and perhaps answer a final question after I have told you something about a few of the core ideas in Gould's evolutionary theory, and told you also something that happens in these last 10 or dozen years after he wrote his magnum opus. Well, let's start. Let's start, of course, with Charles Darwin. Charles Darwin, in whose theory, in whose major work, there is essentially what Gould stressed as a theory of microevolution. The day after day, generation after generation change in populations. And as you know, this change happening in population was explained by Darwin, especially, if not, but not exclusively, but especially in terms of selection. Natural selection later enlarged to encompass also what is called the sexual selection. What is important for the topic of this presentation is that essentially selection is something that happens in population, but where the unit which is selected is basically the individual organism. And this is true in the more obvious context of natural selection. The usual example of Lamarckian origin, but reinterpreted in Darwinian terms, of giraffes evolving longer necks because of the selective advantage obtained by individual with longer neck in obtaining food, and eventually leaving trace of this trait insofar as the length of the neck is heritable. But also, if this should be interpreted in terms of effect of sexual selection, like competition among male deer, for example, using antlers in that case, using necks in this case, eventually what the unit selected for elongation of the neck is anyway the individual. But there are problems in this context, because this theory has some implications, none of which can be taken quietly as granted. One is that the first element is, of course, if we are speaking of individuals, we should have some way to identify individuals among animals and plants, among the most diverse kind of biological things around us, but it's not always true. And other points we will go back to in a few moments. Let's start with individuals. Of course, if we are speaking of human beings, of our more cherished animals, mammals generally, not necessarily pets, but also some less friendly organisms like mosquitoes, but not necessarily limited to animals, because every tree we see in a wood or in a park are usually something that could respond to our naive pre-scientific notion of individual, but things are not always so easy. For example, we have these kind of things. Of course, we call them monsters, but is simply a label, and we can rule out their physical existence around us. One or many. Of course, questions are difficult. In this case, we usually describe this as one calf with two heads. But in the case of corresponding events affecting the human species, we are inclined to speak rather in terms of two partially conjoined individuals. So even if in terms of development, in terms of anatomy, the two objects before us are not that different, our way to classify them in terms of individuality are not the same according to the kind, human or other animal. Of course, things are a bit more relaxed when instead of having conjoined twins, we have physically separated twins, as sometimes happens. I am speaking of identical twins deriving from the same fertilized egg. This happens in several hundreds in our species, but it is the rule, the nearly absolute rule among other species. For example, the 9-banded armadillo, in this case, every female gives birth to a set of 4 identical twins, regularly. And even more conspicuous are these events among some tiny wasps, like the one you see there on the bottom, so small that a female can leave her whole progeny inside the egg of a butterfly. The white thing is a butterfly's egg, but eventually from the early embryo deriving from a wasp's egg, many hundreds, even 1,000, 2,000 embryos derive. And eventually, all those tiny wasps around the dead corpse of a caterpillar are identical twins. What about individuality among coral? Is each polyp an individual, despite the physical, the histological, the material links linking all of them together? Or do we prefer to say that the whole branch structure we see here is the individual, and in this case we lower the level of the polyps to that of parts, of organs? It's a matter of words, a matter of vocabulary we use in this kind of thing. And let me finish this gallery of strange things defeating our efforts to cut them in terms of individuals by showing you the picture of a newly described worm. This is an annelid worm, a segmented worm, a marine animal living in the pores of sponges. And you see there is a main body axis. To the right upper left there are 2 eyes identifying a conventional head, so we see with the mouth. But along the trunk there are several branching, and the gut is also branched, and there are an anus and a pore at the end of each of those branches. So, how many individuals? Difficult to say. Of course, we have in biology something more than pure morphology, pure anatomy, as a criterion for identification of identity, but this is sometimes misleading, as René Magritte suggested many years ago. Of course, we can use perhaps the criterion of, say, the chemical self-perception. That is that molecular identity that is a challenge every time you try to graft an organ from originating from one individual into a physical distinct one. And we know, of course, the long story, the long, interesting and eventually positive story of human action in human efforts in using grafts to help human individuals to survive. But there seems that biological individuals, at least invertebrates, it is so clear, seem to have an internal marker, chemical markers of the individuality. But have a look at this, this system. Again, vertebrates, fish are vertebrates. You see a bigger animal, a female, and two smaller ones. One is, you see distinctly the one with the arrow, it's a tiny male. The other on the right is quite probably another male. These are abyssal fishes. When they eventually, a male and female eventually meet in the total darkness of that environment, they say it would be wastage of time, wasted opportunity if you don't remain joined for life. And eventually what happens is that the two, or three in this case, but the male and female develop a physical continuity. The tissues, the vessel, the blood become one single system, like a modern fetus in mammals. So what about the immunological self? How can these individuals remain glued together without any reject? These are problems. Another kind of problems with the individuality are presented by those animals which, along the life, of course, by saying along their life, I already imply some kinds of definition of individuality. Anyway, animals such as caterpillar and butterfly, for example. Of course, we say they are just stages along the ontogeny, the individual development of one organism. But things are sometimes quite complicated and tricky. Imagine the case of this marine worm, which as conventional adult, is the ribbon-like animal you see on the right, but these animals go through a larval stage, the palladium, the tiny jelly-like organism, of which only a small part, the part, darker part, less transparent part to which the arrow points, only that small cluster of cells will eventually give rise to the conventional adult, to the ribbon worm, whereas all the other parts will simply decay, will be destroyed. So why do we say that there is a continuity, that the one is the larva and the other is the adult of the same individual, rather than saying that the one, the jelly animal, is an animal of which the other is the progeny? Why not speak of different generations alternating along the history of these animals? It's a matter of convention, matter of tradition. We could say, but wait a moment. Even before speaking in terms of genome, in terms of genetic information, there has been several efforts to try to define a criterion of individuality to apply even in this case where different shapes, differently organized forms appear along the life cycle of a given animal. And it was Thomas Huxley, a few years before becoming the strenuous defensor of evolutionary theory, who said that, well, despite the big differences separating a caterpillar from the butterfly, all of these stages and the intervening pupa are ordered starting from a peculiar minimum level, which is an obvious conventional origin of this development, which is the egg. So all those stages proceeding from the egg up to the point where the animal, eventually winged butterfly, will produce again on its turn new egg. These are simply stages along the history for one individual. This can be perhaps one condition. But there are multicellular where there are no eggs, no condition, which are not, where the multicellular is not derived from one egg, but from the coming together of formerly individually living cells, which, so to say, decide to spend some time together, like in the case of these so-called cellular slime molds, where in unicellular stage, like on the top left, after communicating to each other and forming a multicellular mass, eventually give rise to very precisely defined multicellular shape. This is a way to obtain a kind of physical individuality starting not from the egg, but from the multiplicity of individual cells. And in the meantime, in all these, along this process, there are no multiplication of cells like the one obviously occurring from the egg up to the adult. Well, another point which is a bit of problem in respect to a literal interpretation of the blueprint selection of individuals according to the traditional Darwinian tradition, so to say, is the problem that is a problem to which Stephen Jay Gould paid much attention. One of the key points in his theory evolution. It is one of the points around which he developed many very lively opposition to people defending alternative views. Two concepts are contrasted here, that is interaction and inheritance. Of course, inheritance is a key aspect of this theory, because if traits eventually giving advantage to an individual are not heritable, they will leave no trace. They will know this advantage conferred by non-heritable character would not contribute. It will not leave a trace in the following generation. So inheritance is important. It's something on which changes are grafted, so to say. The other point is, however, that without interaction there is no challenge, no selection, no comparison. So which of these two aspects is most important? Which one is the aspect that can define the unit of selection? We can say, from one point of view, very simply speaking, well, in competition for given resources, it's the individuals struggle one against the other. In this way, it is the interaction between the individuals and in respect to the other components of the environment, which stresses the importance of interaction as a key to identify these in this case, the individuals as units of selection. On the other hand, you could say that what matters in the end is eventually what is left. And what is left in the following generation is what is heritable. So these two men, Richard Dawkins and Joseph Williams, developed in the late 60s, Williams in the 70s, Dawkins, the theories of evolution. Both of them eventually stressed the importance of inheritance rather than interaction as the key element around which to define the units of selection. It was particularly, say, vociferous, so to say, Dawkins, who in his first book, The Selfish Gene, the well-known title, already sells the main message that what matters as the key unit of selection is eventually the gene rather than individual. But in this book, what matters here more is that he developed this distinction between units which can replicate themselves, that are units of inheritance, and eventually these are genes rather than individuals. But the genes are not living in a virtual, in empty space. They are not interacting directly among themselves. They are carried by vehicles. And these vehicles are the individuals in whose genome they are placed. So it's eventually the individuals which are as vehicles of genes, which are the interactors, but if the units of evolution are mainly units of inheritance, it is the genes which are the real core of the selective process. These are views to which, with different argumentation, with different logic, both Dawkins and Williams eventually went, at least in a first phase of their speculation. Eventually, both of them revised in very complex way. We have no time to go into details, but this is something to which we'll devote much critical effort, and many of the pages of this magnum opus, which has been mentioned already this morning, and to which I will come back in a few minutes. Well, so genes eventually as replicators, as the most important, if not unique units of selection, as those the very actors in evolution. But let me here start citing some sentences from these last and most comprehensive work left by Stephen Jay Gould. He said in this book that the efforts of Williams and Dawkins to construct selection processes as inherently reducible to selection at the level of gene are based on a concept by which what matters is the way where the advantage or this one, the fitness eventually obtained by interaction of organisms and environment are registered. Of course, what genes register eventually, genes if transmitted to the next generation, are the way in which the account of the success, relative success in the former generation, are recorded and forwarded. But this is a point strongly stressed by Gould. This is not the level at which event actually happen. What happens is interaction, and this brings us back mainly to individuals and perhaps also to other levels of selection, other units of selection. So let's stress this aspect. Selection is essentially matters of interaction, and even if what eventually will go to the next generation will be the genes of mostly the genes of those who are higher fitness, who are in a sense, in very roughly speaking, the winners in the competition. But this view of selection as something essentially working in terms of interaction, something that brings us to consider not simply the individual but also other levels of selection. And this is a field in which one of the author of this theory, which is, we have already seen him speaking of other aspects of this theory, which is Niles Eldredge, contributed a lot. He even wrote a book about this. So I will simply mention this aspect as important, but I will left it out of detailed analysis in this short presentation. Well, critical, say, footnote, which opens up a whole field of consideration, is this very, very careful, very thoughtful remark by Gould. There could be, one could imagine, ideally, say, a limit condition in which eventually genes as the carrier of inheritance and individual organisms as interactor would be eventually the same. One level could be operationally identical to the other. But this would be true only if the correspondence between what we call the genotype, the collection of genes, or in other parlance, the information carried by the genes, would be translated in a very straightforward, direct, and strictly deterministic way into the phenotype, that is the characters of the individuals. Where this correspondence straightforward, simple, linear one, in that case it would be futile. It would be not worthwhile to distinguish, to fight. I am for the genes, I am for the individuals, but only in these conditions, which are very far from being realistic. And I hope in here another page about, say, modern biology, which show how important were these was this thought by Gould, and how the development of these last decades would perhaps have invited Gould, if he had time to revise his view, to incorporate these things. I introduce here something which is, in a sense, something between the gene, or the complex of genes, and the individual. There has been a lot of discussion in the last 10 years or so about modules which are, according to different point of view, functional, operational parts of individuals, either in terms of development, and perhaps Gerd Miller will bring back our attention to this point this afternoon, or unit around which selection operates, which are, in a way, some more integrated part of the ontogeny and also of the interactive history of the organism when placed in its environment. And according to people like Gerd Schlosser, who will be here these days as a teacher in a spring school, perhaps it is at this level because these are set of very integrated parts of the structure of the organization of the organism that we should perhaps pay more attention. It is perhaps at this level that selection operates more intensely than at the level of either the gene or the conventional organism. This is another, say, way to introduce something which is very important, which Gould paid increased attention towards the last few years of his life. That is the importance of development as something to be studied as complementary to the study of evolution. But what has been a very fashionable aspect of very topic of much attention in the last few years is what has been called the genotype to phenotype map. That is the very diverse kind of correspondence, very diverse kind of relationship by which what conventionally we say is written in the genes, so to say, is eventually to be found in the ancient phenotype, in the structure of an embryo, of a juvenile, and eventually of the adult. I mention here vessels, perhaps our human vessel, veins, arteries, because this is one example where we know that genes determine more or less, well, a vessel is made this way, branching of vessels are made this way, this is an artery, this is a vein, but that's all. The detail of the branching pattern are not described in genes. It's something that happens during the individual development. If you look carefully at the pattern of your blood vessels, looking comparatively left and right, you will see big difference. Every individual is asymmetric, and there are of course not a description in the genes for the right and the left hand. Check, please. Gianna, you agree? And again, development becomes more and more important. For example, these two men, philosophers deeply interested in biology, and also co-authors of theory, which is important in the field of this borderline between evolutionary and developmental biology. Paul Griffiths and Gray, in a paper written many years ago, 94, stressed the point. Let's try to understand what is really replicated in development. It's not strictly matter, or say, it's enough to consider the genes as the seat of bookkeeping, as Gould said. This doesn't explain straightforward what eventually we will find as the result of development in the next generation. Well, one of the main points calling attention in this field is so-called phenotypic plasticity. Phenotypic plasticity means that same genes, different phenotypes. Same genes, different phenotypes, depending on different environment and condition. For example, among these aphids, these small insects, we can have both winged and wingless individuals according, for example, to the length of the day. Generations growing, developing under short day or under long day condition, either develop wingless or respectively as winged forms. In this case, and in a few other butterflies, you have different seasonal generations. So these are not two species, but simply the spring and the summer generation of the same animals. So the offspring of the lighter population, lighter generation, is the darker form. And the offspring of the darker form is again a light form. In this case, it's again partly day of the length, partly temperature, determining what happens in development, eventually giving rise to different forms, different phenotypes, with the same genetic endowment. And again, even something which seems to us to be so fundamentally genetic, like sex, can be determined in some animals simply by exposure to different environmental conditions. In many reptiles, for example, in the alligator, it's the temperature at which a critical phase of the embryonic life is spent that determines the sex. Lower temperature male, higher temperature females. In other species, different. But it's not written in the gene, not written in the DNA. Or the length of the horns in these dung beetles. It depends on the amount of food obtained by the larva. No genetic difference among the small horn and the long horn kind. And they are not intermediates. There are two distinct forms, alternative forms. Or again, among social hymenopterans, social insects like ants. For example, the difference between a queen, a mature, reproductive mature female, and a worker, a sterile female. These differences are simply nearly always due to condition or the quality of food they got when larvae. And to finish with this series, it is crowding condition, the physical stress of being in contact with other individuals of the same species, which determines the origin of the migratory gregarious form in these locusts. Otherwise, when living, say, in less crowded condition, they grow into the traditional solitary and sedentary form. But the borderline between what is genetically controlled and what is environmentally controlled is sometimes very, very thin. This is a wonderful example studied recently in the pea aphid. Again, aphid, again, winged and wingless form. But in this particular species, in the female, getting wings or not depends on environment. In the males of the same species, same population, same animals, but in the males, getting wings or not depends on having this or that alternative form, this or that allele of one specific gene. Interesting, but it's not the whole story. The whole story is that also in the female, the environmental influence, so the length of the day, is eventually, the effect of the length of the day, is eventually mediated by the molecule, the protein, which is expressed by the same gene, which in the male, when present in one or the other form, determines the wing or not. So eventually, the same gene is involved in this process, but in different way. Autonomously in the male, or through having the switch moved by environment in the female. So the borderline between what is phenotypic plasticity, what is genetic polymorphism, is thin, and we are speaking of evolution, something which may change rapidly in the history of a population, of a species. Another point which is very problematic to identify an organism with a genome is that everywhere we find multi-genomic system. Some of them are very traditional systems, like lichens. Every lichen, and there is a very strong specificity in the shape and the ecology of these forms, which are the result of association of a specific fungus species and a specific alga species. But also we have other systems like those mycorrhizae that between roots of trees, of different plants, and again fungi, or the root nodules, a result of interaction between a specific kind of bacteria and the roots of legumes, or the galls was a strong specificity is in the sense, let me use here a word, a term coined by Richard Dawkins, why not? The extended phenotype, something which is outside the body of the organism which has a given genome and eventually is expressed. Because a gull is physically a part of the plant body, but is largely conditioned its shape by effect of something which is dependent on the insect which stuck in the plant tissue. And a wasp and the plant interact, eventually giving rise to those very specific kinds of galls. And the final example are we humans. In our gut, as like in those diversity of organisms, we have what we call often our gut flora, gut microbic flora, which are basically a diversity of bacteria. And the relationship between our system dependent our organism is dependent on our own genes, our nuclear genes, and what is offered by these bacteria is a kind of symbiotic interaction, which is hardly less strict than the one of lichen. The last point in this analysis of implication of selective theory of microevolution is that, strictly speaking, in Darwinian terms, focus is on individual organism. But along these 150 years, there has been a long history of people looking at possibly different levels of selection. The easiest way was to look inside the organisms, like this German embryologist Wilhelm Roux did around 1880, when writing this book on the struggle of parts within the organisms. Parts like molecules, cells, organs. And what is a modern edition of this view, the well-known neural Darwinism developed by Edelman, considering strong competition among our neurons during development. Well, back and finally to the last section, to Stephen Jay Gould and his efforts to complement the traditional theory of microevolution to a view encompassing aspects of macroevolution, to which several of people in this room, some of people in this room, have strongly contributed. But let me consider something outside the Gouldian circle for a moment, for the last moment, mentioning this man, also somebody interested both in practical study of biology and at the same time in theorizing, Liubas, who developed an interesting study of individuality, it was the model of individuality, stressing several aspects, which are mainly of dealing with development. I cannot go into detail. And eventually, a contribution by a man, David Hull, a philosopher of biology, who died 1 or 2 years ago, who in an early paper of the 80 stressed an important point, which is important in our context of moving from micro to macroevolution. That is that there are individual is not necessarily the individual organism. We can conceive of individuality of a system, particularly of biological systems, hierarchically ordered below and above the conventional organismal individual organism. And as long as this system have individuality, the problem is what individuality is, but eventually along this story of system getting individuality along with the history of things, history of life, new levels of selection eventually emerge. Another contribution of this more recent contribution by Richard Michaud stressed kind of what were now more or less popular as evolutionary transitions. transitions, of which the transition from single cell to multicellular organism is just one, but there are something below it and at least one above it, the transition between solitary organism and colonies. Think of ants, of bees, of humans. But it's not the whole story. This is a hierarchical view, but it's not the whole story. Of course, Gould was very well aware of the difficulties presented by adversary of this hierarchical and multi-level model but he, in his book, he argued very carefully to demonstrate the relevance of the multi-level model, even despite these individual arguments. And especially the last one, which is interesting. One could say, well, there is selection, say, at the level of species, but species are eventually made of individuals. So why cannot we be happy with describing everything in terms of individual selection? Why do we need to distinguish another level? The problem is that levels can have peculiar characters. And a couple of people I will mention, he will say that along this hierarchy of levels something peculiar happens. This was a very tricky question for Gould, and there are a couple of very interesting pages, both from intellectual and from the personal level in his big book. Let me read with you some of these sentences. No other subject in evolutionary theory has so engaged, at the same time confused me throughout my career as the definition and elucidation of species selection. For no other problem have I undergone so many changes of viewpoint before settling on what I now consider a satisfactory framework. I made two sequential errors of opposite level. A couple of minutes and we finish. Thank you. Even many before, Ronald Fisher, when writing the second edition of his book on the genetical theory of natural selection, have demonstrated that in terms of models, say mathematical models, there could be space for different levels, and especially this species selection. They said, in fact, perhaps there is no evidence. I don't know of any evidence. The problem was discussed again, for example, by Dick Lewontin. We said that there is a plurality of levels. Let's just investigate carefully nature. There will be interpretation, last two lines, interpretation of the principle of selection could be made for species rather than population, and even communities rather than species. So, and somebody here in the audience has been repeatedly called as a colleague, as a helper, as an inspirer, as an adversary sometimes in Stephen Jay Gould's pages. And said, when Niles Eldredge and I first formulate a punctuated equilibrium, I was most excited by the insight that trends would lead to be reconceptualized as a differential success of a species rather than anagenesis within lineages. I then committed the common fallacy of extending personal excitement far, and I made the error of labeling as species selection any pattern that needed to be described in terms of differential success for species treated as stable elements. In other words, we failed to distinguish selection from sorting. But later, it was the time of the interaction of Stephen Jay Gould with Elisabeth Lloyd. In this time, say, in reaction to this excess, I retreated too far in the opposite direction, by restricting species selection to cases characterized by emergence of a new character at the species level. But eventually, another lady here in the audience, and next on the floor, yes, Stephen Jay Gould's interaction with Elisabeth Lloyd convinced him that the emergent characters, while properly identifying species selection, only identified a subset of genomic cases, but species selection is much more general, much more pervasive, and it's enough to be able to identify emergent fitness as something that characterizes this level of individuality, as the level of species, as a peculiar unity of evolution. Well, let me finish. I come back to my introductory words about attitudes of people in respect to evolution, of this problem of evolution. I mentioned the contrast between very few, indeed of Italian academic world, attending a big evolutionary congress, and many, many millions of people interested in or at least ready to listen to talks about evolution. There are different kinds of people involved in evolutionary biology. I mean, scientists, researchers, theorists like Joe Smith, people working in the lab, mostly with molecular tools, people working in the field, collecting, studying population, studying adaptation in the field. You have paleontologists, different kinds of people. To have a perception and perhaps a numerical guess of what people do, how much people are still interested in the question I have been presenting here in this talk. Let me give you an example. I browsed through the programs of the two last editions of the big evolution meetings held in America each year. And the last edition were attended by, at each of these meetings, were present some 1,500 between talks and posters at each. So, browsing through 3,000 titles between the two editions, I found only 3 titles mentioning one of the concepts I browsed together from the big book of Stephen Jay Gould. Nothing, no contribution about species selection, no contribution about hierarchy of levels in evolution. Only 3 titles mentioning more or less multi-level selection. Does this mean that people say, this large community of evolution biology is not interested in these things, or vice versa, on the other hand, that the theory has not need of the results deriving from the different corners of this world of students? I think that the end brings me back to something that Dick Wanting said in his introductory talk. That is, studying evolution means to do something in detail, different level, working with molecules, working with population, measuring with fossil, doing the painstaking daily work with any kinds of things you may prefer to look at, and at the same time it is speculating about the general aspect, the big problems, big trends, big rules of evolution. We need both things. And in this case, in this sense, Gould gave us a big example, and we need both to read and comment, annotate his book, bringing in the contribution of ongoing contribution of research, but at the same time we must use in our daily research as paleontologists, as molecular biologists, as systematists, phylogeneticists, and so on, interested in evolution. We must bring in the big vision of some visionary great man as Stephen Jay Gould. Thank you.